I'm going to finish by zooming out to the whole brain. So just as we want to go from the restricted view of the lab to the real world, we want to go from the narrow view of one structure like the amygdala, about which now we understand a lot. We can record single neurons in the amygdala in neurosurgical patients who have autism. And we want to acknowledge that, in fact, the amygdala is plugged into the rest of the brain and part of this big network. So can we study patients who have lesions in all of these different parts of the brain? You might think, well, that's next to impossible. It turns out to be possible in collaboration with where I was prior to coming to Caltech in 2004, where I did my postdoc and was junior faculty, which is the Department of Neurology at the University of Iowa, where back in the 80s, Antonio Damasio and colleagues established a large registry of neurological patients that have lesions in different parts of the brain. Some of them have amygdala lesions, some of them have lesions elsewhere. There are over 1,000 of those that have been studied, and about 500 or so that are currently available. And so we can study these patients and try and correlate their lesions with aspects of their behavior and cognition. And just to give you an overview of how these lesions are sampled, so if you take various slices of a brain and you depict how dense the overlap of lesions is by the colors, this picture just makes the point that most of the brain is covered in this registry of lesion patients. They're not homogeneously distributed, as you might imagine, so they tend to be, as these hot yellow colors illustrate, they tend to be more lesions in certain parts of the brain, because these are vascular territories where if you have a stroke, you would have a lesion. So not all parts of the brain are lesioned with equal probability, but nonetheless, they're quite well sampled. This is an example from, I think, 400 subjects whom we studied. The logic is straightforward, so it's just an extension from case studies like we looked at in the case of the amygdala. So if we look at performance, say ability to recognize facial expressions of emotion, in healthy individuals, in a group of those, you would have a histogram, hopefully roughly normally distributed, something like this, in an idealized case. Some people perform really well on the right, some people perform a little worse. There's some spread, some variance in the mean, and so that's how it would look. And then the classical approach, which is what we took with this patient with amygdala lesions, is you have a subject, a patient with bilateral amygdala lesions, who's way outside of this, and then you could put some statistics on that. But if instead you take a group of 100 or 200 people with lesions all over different parts of the brain, what do you get? What happens to this curve? Well, it shifts like this. So on average, brain damage doesn't improve your performance. It makes you variably worse. So some people are pretty bad on any given task, and some people are unaffected. And the degree to which they get worse or are unaffected depends presumably on where in the brain they have the lesion. So you could simply partition this, for instance, and ask, where do all the lesions overlap of the folks who are unaffected on this task with their brain lesion, and where do all the lesions overlap of the folks here? And that would show you which parts of the brain, when lesioned, are necessary for that task. So that's the rough logic. You can do a more continuous version of this, correct statistically for all the multiple comparisons between the different parts of the brain that you're looking at, and you get data something like this. We've done a series of studies like this now with my former postdoc Jan Glescher, now faculty in Germany, capitalizing on this large lesion registry. So in this particular example, we looked at general intelligence, Spearman's G, and we asked where in the brain do lesions impact general intelligence. And these hot regions that are shown here on the brain are regions within which lesions impact general intelligence. It looks very scattered, as you would expect. There's no single clear region that's responsible for intelligence. Instead, there's a whole bunch of scattered regions that are all interconnected. So it's a very diffuse system. And presumably, that kind of diffuse system is something that we will find also in relation to social cognition and social behavior. Many different parts of the brain that are interconnected. And that raises the point that we can't just think of these brain regions in isolation, but we need to think of the stuff in the middle, the white matter, all the cabling that connects them, which brings me to the last disorder that I want to tell you about. So if you take a human brain, shown on the left here, and you carve away at the top the cortex and look inside, what you see is the fact that most of your brain is not cells. Most of your brain is connections between them. Most of the brain is white matter that connects all these different brain regions. For your brain to function, it has to be connected up in the right way. And what you see revealed here in this top-down view, this big bundle here, is 200 million connections, 200 million axons that connect the left and the right cerebral hemispheres, the corpus callosum. This is what's sectioned in so-called split brain patients surgically for the treatment of epilepsy, extremely rare. But there's a population that we've been studying in my lab, which, like autism, is a developmental population, and these are people that are born without this corpus callosum. It's extremely rare, but 1 in 4,000 or so. But using the appropriate exclusionary and inclusionary criteria, you can select high-functioning adults with agenesis of the corpus callosum. So these are people with IQ in the normal range that developed without this massive white matter connection. And here's just an MRI scan. So here on the left is a healthy brain. Here's the nose, eyes, mouth. And this sort of semicircular shaped white thing up here is the corpus callosum caught in cross-section and is missing completely in the brain of a patient with agenesis of the corpus callosum. Similar things shown here using a technique called diffusion imaging, where we can look at the direction of connections in the brain. Healthy brain on the left, red denotes fibers that run from left to right. So this big red thing in the middle here is the corpus callosum that connects the left and right cerebral hemispheres, and it's absent. There's nothing connecting the left and the right cerebral hemispheres on the right in a brain of a patient with agenesis of the corpus callosum. Well, so how do these patients present? They're, by virtue of how we select them, they're high functioning. Many of them have jobs, have families. They all have IQ in the normal range. If you do neuropsychological assessment, you find that they have an interesting profile of peaks and valleys in what they can do. They're often pretty bad in sort of very flexible, unconstrained kind of processing. And they're pretty good on sort of rule-based, more rigid kinds of processing. But the most relevant thing is that about a third of these meet formal criteria for an autism spectrum disorder, and essentially all of them say things that sound very similar to what high-functioning people with autism would say. So if you just ask them, what do you have difficulty with in everyday life, they will say, it's very easy. I have difficulty with other people. I can't figure out what people are saying. I often say the wrong things. I have difficulty maintaining friendships. I have a hard time maintaining my job. It's all about social interaction. They say exactly the same kinds of things that many high-functioning individuals with autism say. So we think here we have a neurological model for at least one aspect of autism, which is the social difficulties, that arises because you've disrupted the ability of all these different parts of the social brain to communicate normally with one another, like language processing in the left hemisphere, more emotional processing in the right hemisphere. In order to interact with a person, when you're talking to them and looking at them, you have to be doing both of those at the same time, in real time, really rapidly. And your brain can only do that if there's a very fast pathway for communicating between the left and the right hemispheres, and that's missing here. Just to finish up, there are a couple of very exciting future directions for this and reasons we're particularly interested in this disorder. One is that there's a mouse that also has agenesis of the corpus callosum, this particular mouse strain, BTBR, and what's just shown here in green is the white matter connections in the left and the right cerebral hemisphere in this mouse brain. Normally, this would be going across, but the mouse has no corpus callosum, so this is a mouse with agenesis of the corpus callosum that we've been imaging at the Caltech Brain Imaging Center. Turns out these mice also behave strangely in their social behavior, and in fact they're being studied as a mouse model of autism. Now, mice don't have autism, but this particular mouse model shows certain types of behavior that people think is a model of aspects of autism. Certain kinds of grooming behaviors that look like abnormal stereotype behaviors, abnormal maternal behaviors and separation cries, and so forth. There's a final reason that we're very interested in this disorder and trying to study it very early in life, which is it's pretty hard for anybody to see this unless you're an expert. But this is an ultrasound of a pregnant mother showing that the fetus has no corpus callosum. So agenesis of the corpus callosum is now regularly diagnosed in utero. And so you can imagine that you would know that this child will have agenesis of the corpus callosum before birth, and you can start studying the development of the brain and the development of behavior already at birth or before, and you will know that a third of these folks will go on to develop autism. And that's something that we can't do in people with autism yet, because you can't diagnose autism until about age 3. So these are the conclusions, just to summarize again what I had up in the very first slide. And I finished with just a couple of sort of broad open questions to finish up here. So one important point is that development doesn't stop when you're a child or an adolescent. In fact, it never stops. And so autism is a developmental disorder. Agenesis of the corpus callosum is a developmental disorder. And we need to understand these not just as disorders of childhood and infancy, but disorders of lifespan development. Your brain is changing all the time throughout your life. And understanding how the brain changes in people with autism is a huge topic, in particular how autism and aging interact, as I mentioned. There's another big, very challenging topic, which is the question, everything that I showed you is about faces and social stimuli, but you could and should ask the question, well, how specific is this to the social? Maybe they just have very general, people with autism just have very general processing deficits, or people with amygdala lesions have very general deficits that might be most apparent in the case of social stimuli, perhaps because they're more complex or something like that, but there's nothing specialized for social cognition. To do that, we need to have appropriate controls, and so we have lots of controls that we're devising all the time that try and match social and non-social stimuli and tasks in all possible dimensions we can think of to try and show that there's something specific about processing social information as such. And then finally, we want to be able to link all of these different levels of analysis, which of course you can't do in a single lab, but there are various large consortia for doing so. But the last question, very last question to finish up with, is this one here, which I think is a really important question, and it's not just philosophical because it has practical importance, which is what is autism? And more broadly, how do we think about and how do we characterize individual differences in everybody? So all of us are different in our social behavior. How do we characterize that? What are the basic dimensions of social cognition and behavior? And we're fairly far behind there, but there's some very interesting developments very recently for thinking about this. If you go back to the 1800s, when phrenology was started by Franz Joseph Gall and his followers, there was this picture of the relationship between psychology and the brain that we can sort of laugh at these days. The phrenologists thought that all of these different faculties indicated in the little boxes here could be mapped onto particular parts of the brain. So it doesn't work quite like that. We now know that a lot of these you can't map in a clean way like this. We know that many functions are distributed in networks. But the phrenologists had the basic idea, right, which is that there's specialization of function in the human brain. And the question is, what is it that's specialized? It's pretty easy when you're talking about vision. So vision, is sort of back here. Hearing is sort of here. But higher cognitive functions, well, the phrenologists had this whole list of them here. And many of these we can sort of laugh at now, but many of them are still perfectly respectable. So color vision, language, memory, these are still terms that we use today. Many other ones, I don't even know what this would mean, comparative sagacity, poetical talent, etc., we can laugh at. But I think we should seriously question all of the categories that we currently use. So we can talk about systems for fear, sadness, attention, memory, decision-making, all of the terms that psychologists use. But are those the right terms in terms of the biology? Is that how the brain really works? And this is relevant to understanding autism because at about the same time, just before the DSM-5, the latest edition of the Bible for diagnosing psychiatric disorders was released, Tom Insel, the director of NMH, made the following comment. So he said that basically these DSM diagnoses, the diagnoses for psychiatric illnesses, are just based on a consensus. So psychiatrists that sit around a table and decide that these are the criteria for autism or schizophrenia. And they're not based on objective lab measures. And so he concluded by saying it's critical to realize that we can't succeed as scientists if we use these DSM categories as the gold standard. The diagnostic system has to be based on the emerging research data, not on the current symptom-based categories. But this doesn't mean that the current diagnoses offered by DSM are useless. If we didn't have those, all hell would break loose. So we need some consistent criteria, and they do measure something. Clinicians are picking up patterns when they diagnose autism or schizophrenia, but what they do is diagnose this just through behavior. They just have an interview, they look at the history of the child. It's not based on genes, it's not based on brain scans. And so Tom Insel's call here is, at least as scientists, to try and have a somewhat more basic vocabulary out of which then hopefully we could assemble these disorders, but not to start with the disorders as biological entities, because he just doesn't think that that's how things will play out. What's the alternative? Well, there are these so-called research domain criteria, which NIMH is now pushing. So if you're writing research grants to be funded by NIH, you have to pay attention to these. And reviewers are supposed to not look favorably onto grants that just stop and say, we're looking at autism or we're looking at schizophrenia. You should be looking at particular domains like fear, approach motivation, attention, etc., that would cut across all of these different disorders. So these might be variably impaired in autism, in schizophrenia, in mood disorders, and cut across all of them. I'm not going to say much about this other than just the big flavor. So what people are doing now, the latest sort of push, has been indeed to find maps of brain activation that relate to such simpler building blocks out of which we could assemble these symptoms like autism and schizophrenia that are currently diagnosed. So for instance, emotion, valence, arousal, that maps onto this particular activation pattern, regret, surprise, and some others onto this, and so forth. So the idea is to come up with a more basic vocabulary that reflects the underlying brain system, the actual biology, and presumably the sets of genes out of which we can then construct these categories. And people have been trying to do that. So in this particular paper here from one of the people who's been leading this, Russ Poldrack, what he's done is taken those patterns, those simple patterns of brain activation, not for autism, not for schizophrenia, but for more basic processes like fear, attention, memory, many of them, gleaned from over 6,000 research studies. So he did a big meta-analysis of all of those. If you cluster all those brain activation patterns, you find that you get groups that do make a lot of sense. So there's mood disorders here in red. I know you can't understand any of these abbreviations here, but these are mood disorders. This schizophrenia is over here. Autism is in blue over here. So you are able to reconstruct these diagnostic categories to some extent, but you don't start with them in the first place, but you start with something that's much more agnostic. So that's the future. That's where currently things are heading. It's still very early on. We know a lot, and there's a lot of big research efforts underway to dissect the details of the brain and to map out how different parts of the brain are connected. What we're really still lacking is an analogous thing for the mind. We don't know what the building blocks are psychologically onto which we should map those brain data. So that's what people are trying to do here in order to ultimately make a tight link, much tighter link, between the biology and cognitive functioning that can inform psychiatric illnesses. So let me end with that by acknowledging a lot of people in my lab whose work has been reflected in various ways in the slides that I showed you, collaborators, the University of Iowa for the lesion patients, these hospitals here where we do the neurosurgical recordings, and then of course funding sources, both federal and various foundations, the Moore Foundation, which gave rise to the Caltech Brain Imaging Center that you'll see next to make possible all the imaging. And if you want more information, there's our lab website here, a graduate program that's relevant here at Caltech, and the website for our Conti Center from NIMH that funds a lot of this work. And I'll end with that. Thanks.
I'm fascinated by the fact that a lot of the autism people and the amygdala looked at the mouth. I mean, that's not an irrelevant area like the chin or the forehead. So I was wondering, have you been able to detect in smaller children, infants, that might be a learned behavior, that they're not getting information from the eyes, but you can actually get a lot of information from the mouth, especially auditory and tone of voice, volume. Yep. And I'm wondering if that could be some way that they're compensating and learning to get information.
I do think that that's a reasonable hypothesis, yes. You would imagine that if you're not, I mean, it's a question, why are you not looking at the eyes and why are they not salient? There's several possibilities. One is that you just don't find the eyes attention grabbing and you don't look there. Another one in autism is that you find the eyes actively aversive and stress inducing, and so you actively avoid them. Possibly both of these happen. But then indeed, as you say, if you're not using the eyes and you're trying to figure out information from a face, what's the next most informative region? And it's the mouth. And so indeed, you could think of this as a compensatory strategy that people with autism have developed that's reflected in some of those other pictures that I showed you where we have the experiment with the little pieces of faces, where indeed we find that high-functioning people with autism make less use of the eyes, but they make exaggerated more use of the mouth region of the face in order to compensate. And it's worth noting that on that task, so they get exactly the same task as the control group. It's a very difficult task. You see little pieces of faces. You have to push buttons. And their accuracy and their reaction time, so metrics of their performance, are indistinguishable. They're perfectly normal in their ability to detect fear or happiness in the faces. But the strategy they use isn't normal. So they're able to compensate as a consequence. We also see that reflected in the eye tracking data, which I didn't show you details of. But what many people with autism do when they look at a real person, this actor that we had, is that they go to the eyes and they go to the mouth and they sort of keep going back and forth between these two. Our interpretation of that is that people with autism have a propensity and find the most salient to look at the mouth, as what we were just saying. But in addition, they've been told that they should make eye contact when they're interacting with a real person. And so these two processes, a sort of automatic propensity to look at the mouth and then a more cognitive top-down strategy to also look at the eyes, as a consequence of which they keep going back and forth. And if you just sort of lump this together and you ask what's the proportion of time that at least high-functioning adults with autism look at the eyes when they interact with a real person, many of them are perfectly normal. But if you look in detail, they're not normal in how they do so. It's very rigid. They look at the eyes in a very rigid way, and then they keep going back down to the mouth.
Some years ago, radio commentator...
Sorry, where are you? Can you put your hand up? Oh, there. Okay, sorry.
Radio commentator...
I make bad eye contact. The wrong person.
Radio commentator Paul Harvey told of a situation where a magician was performing, and a subnormal youth in the audience would see just what the trick was, because all the other people their attention was drawn perhaps to the eyes and mouth.
Right, yeah. Magicians are capitalized on exactly that, their ability to socially manipulate our attention, and as a consequence of which we don't see what they're doing, and so it's magic. But if you're not susceptible to that, if you're immune to that, then the magic wouldn't work. Yeah.
So, very, very interesting. And one thing I was thinking about from the very beginning of your talk is how much of this differs by culture, because I seem to remember at one point reading something about how we express emotions depends partly on the facial musculature, which can vary across the world, and partly on culture, and how does autism vary in these various countries. And a lot of the research, I know most of the fMRI research in this country, the control subjects tend to be students, tend to be white, tend to be educated, tend to be English speaking. And a colleague at NSF has recently given me a paper that considers these subjects weird, actually. And I've forgotten what each of those letters stood for.
White, educated, industrialized, something, but yeah.
Right. that this actually represents a small fraction of the world population, and yet
right.
all of the conclusions that we're drawing about psychology come from this population. And so I was curious about the cultural differences for autism and your findings.
Yeah, no, that's a really important point. So there are big individual differences. So everyone in this room, if I did any of the studies that I showed you, you would all be slightly different, and some people might be quite a bit different. And we all know people that make a lot of eye contact, and some people sort of look at their shoes when they're talking to you, and they don't have autism. So they're big individual differences, and some of those differences are systematic in the sense that they vary across cultures. Some probably map onto gender differences. Some map onto your level of education and age, et cetera, et cetera. So you're absolutely right. There's sort of two arguments. You could argue that, look, this is so complicated. If I start, accounting for age and gender and culture and everything, I can't do that. So I'm going to pick, just as I pick an inbred mouse strain for my lab experiments, rather than gathering wild mice in the forest, I'm going to pick something that's actually homogeneous and that I can, get some reliable and replicable results with. Caltech students say, right? But they're not typical at all, as we know. And so the question then is, how does this generalize? So I think the approach that many people have taken, which is defensible, is to do this in stages. It's, I think, defensible to say we're going to start with less than the full complexity and not look at culture. But then, once we have results, of course, we do want to study this in different cultures. So there are cross-cultural studies. There are cross-cultural findings for the experiments that I showed you. So people, Asian people compared to Western, have differences on average. They're subtle, they're small, in how they look at the eye region of the face also, and parts of the face that they use to judge emotion. It's complicated nowadays because cultures aren't so separate, and so there's lots of complexity. How psychiatric illnesses are diagnosed across different cultures is yet another layer. And so you would hope that everybody in the world would adopt a single set of criteria, even if it's DSM-5. As long as it's consistent, at least we're all talking the same language. But even if you go to Europe, they use ICD-10. It's similar, but they don't use the DSM. So there are differences, and one big challenge is to try and make this consistent. For research studies, many people have tried to have a consistent set, which is much more than DSM. The Autism Diagnostic Observation Schedule and measures like that Kathy Lord has developed here in the US, that are sort of very reliable research measures for diagnosing autism that go beyond the DSM. And the hope would be that everybody in the research community, at least, not necessarily for clinical diagnosis, but all the researchers in the world would use a consistent set of criteria for diagnosing autism or schizophrenia or anything else. So it's a very important issue and one that needs a lot of attention.
Hi, hello. So I'm wondering if you have any data or have performed experiment on those who have been diagnosed as Asperger's syndrome, which is a less severe kind of autism.
Yes, so in our studies, we typically combine high-functioning people with autism and with Asperger's syndrome on the DSM-4 diagnosis. So typically, we have both of those in there. We don't typically have large enough samples to try and make distinctions between them. So we don't have any particular studies where we're trying to look at differences. And of course, according to DSM-5, those are not diagnosed separately anymore. They're just autism spectrum disorders, although there are scales in the DSM-5 diagnosis for intellectual functioning and language delay that would still give you those distinctions between Asperger's and autism, even though they're not separate categories. But so the short answer to your question is, in our study, all the studies that I showed you combine, according to DSM-4 criteria, high-functioning autism and Asperger's syndrome together. So there are people with both in there. Maybe one last question, or maybe we can take.
So it was tantalizing to look at some of the brain maps and to see these localized and non-localized regions. So in looking at that and understanding spatial data, I was thinking back to my time here as an undergraduate, thinking about how in physics we were taught to linearize problems. In mathematics we were taught to look at orthogonal vectors in linear algebra. And then in PDEs we were trying to understand orthogonal basis functions. So then looking at this three-dimensional data, it makes me wonder, are there ways to map this in terms of mapping onto basis functions and understanding what are things that are truly orthogonal and unique, what are things that are not, either through SVD or other mathematical techniques to try to understand how to map them onto something?
Yeah, definitely the basic idea is that, in assembling this sort of vocabulary of basic cognitive processes and the associated brain systems, it's like doing PCA or something to come up with some kind of basis functions that once you know the set of those, then you can just linearly combine them and you can reconstruct everything that you're interested in. The extent to which they're orthogonal is another question, and they're typically not orthogonalized, at least in terms of the brain patterns. There's lots of overlap. But that may be a question of the resolution we have available with fMRI. So you would have a bunch. So what people often, actually you can pull these networks out just when people are doing nothing. So one thing that's popular to do these days, because you can do it in infants as well, you just put them in a scanner and you have people do nothing. They're just resting in there. And you just collect 5-10 minutes, so a big time series of brain activation. And what you find is that activation in the brain, in different brain regions, fluctuates over time. So you have this time series, and you can simply ask, what's the strength of the correlation between one part of the brain and another? And so you use things like independent component analysis to pull out networks. And so there's a complement of networks that everybody has reliably, no matter what they're doing, if they're just lying in the scanner at rest. If you look at those networks, their actual patterns are all distinct, but there's lots of nodes that overlap spatially. But that may be an artifact of the fMRI. So it's possible if you're able to zoom in and look at single cells, you would find a region that lights up an fMRI in multiple networks, but in fact there are different non-overlapping populations of neurons within those regions that are beyond the spatial resolution of fMRI. So there's lots of problems here, but one big issue is to try and get at these networks in a sort of multi-resolution way. We have fMRI. The advantage is that gives us a picture of the whole brain at once, but it's really blurry. We have little cubes that are about 3 millimeters big. If you want to record from single neurons, like we did in these surgical patients, you can do that, but you can only do it in very small parts of the brain, so your window is tiny. You don't record from the whole brain. So trying to get both whole brain field of view and data at the single cell resolution is sort of the holy grail. Of course, once you would have that, the next question would be what to do with that gigantic amount of data. So perhaps I should give a quick intro to the Imaging Center, or let's do two more questions. Okay, fine.
Where do you see all of this research leading? Does it lead to possibly earlier diagnosis and therefore working, more of the social aspect of working with people with autism? Does it lead to therapies like drugs for schizophrenia? Where do you see this all leading in terms of improvement of the situation?
Right. Those two you mentioned, definitely. So so I'm a basic research scientist. So my goal is just to understand the social brain, and in so doing, then to also understand disorders. But the two that you mentioned, being able to understand what happens early on, so how dysfunction develops early in life, in particular in relation to developmental disorders like autism, that's definitely something that many research groups are working on now. So there's studies, for instance, where you can image infants already at 6 months of age before you can diagnose autism, and you can characterize their brains, and then you wait a couple of years, and you see which of them went on to develop autism and which not, and you ask, was there anything at 6 months that already distinguished those two groups? And so people are finding some very early signatures that is giving us information about which brains are likely to go on to develop autism and which brains not. So a lot of the research effort is focused on early development, and in so doing, once you know what some of the early mechanisms are, then of course you're in a position to intervene. And you could imagine tailoring interventional strategies to particular individuals or particular subgroups of autism. The earlier the better, so that you can change this developmental trajectory. So both of the things that you mentioned. There's a third one that I think is really important, which is not really for the people with autism as such, but for all the rest of us. So if we know better what the challenges are and how the minds of people with autism work, that will educate the rest of us in order to try and provide more supportive, more structured environments in which these individuals can function better. So I think the educational component, by just knowing how it works, is also a big one.
One more question?
In the patterns of watching what's on the face, there was a distinct emphasis on the right eye over the left eye. Is that a matter of just the selection of the thing that you chose to show it, or is that general?
There seems to be a general propensity to look more at the right or the left eye that's reliable within a person. So some people look more at the left, some people look more at the right, and if I did experiments over quite a few days with the same person, on average, they would tend more to look at the left or more to the right. What the causes or consequences of that are, we have no idea. But you're right, it's not symmetrical. The expression of emotions on the face is not symmetrical either. And depending on where you look at the face, there would be a higher proportion of information in the left or the right visual hemifield. So there would be some asymmetry, both in the control of muscles in the face and in your perception of it in relation to the left and the right hemispheres of your brain. The left and the right hemispheres of your brain are going to be processing somewhat different social information. And so presumably this pattern that you picked up on has some consequence, but we don't know what it is. But it has a consequence in terms of the proportion of information that's going to be processed by the left or the right half of your brain. Big unknown territory, though. Okay, so let me give a quick overview of the Caltech Brain Imaging Center for those of you going there next, just so you can see what it is. So it's immediately adjacent here in the Broad Center, so less than a minute's walk outside here. And you'll go inside and see the various scanners, of which we have 4. And right at the beginning, I want to say one important thing, which is not to go into the scanner room. So when you go in there, people will show you things. They may have the door open so you can see the scanner, but don't go into where the big MR scanner is sitting. And the reason for that is that it has an extremely strong magnetic field that will not only demagnetize all of your credit cards, but also suck all the coins out of your pocket and turn them into bullets, which you don't want to happen. So don't go into the scanner room. So what do we have? We have 4 scanners in the imaging center. And so you will see one of these, and people can tell you about the research. This is right outside the Broad Center, so right where you'll walk, there's those sort of sidewalk tiles outside. And this is when we had one of these scanners delivered. It weighs about 14 tons, and it's just being lowered by a crane into the rooms below in the basement, which is where the imaging center is located. So to get these scanners down there, we actually had to open up the sidewalk, get a huge crane in place, and drop these things in. The Siemens technical folks don't like me using the word drop, but we gently drop them in. What they are is coils of superconducting magnet that is sitting in a bath that's very cold of liquid helium. And so this gets charged up with a very large amount of current that, since they're superconducting, continues forever. And as a consequence of it going around this coil, induces a very strong permanent static magnetic field. And so which is the dangerous part of these magnets. And then there's a lot of complicated machinery inside here that the folks in the imaging center can tell you about that allows us to use this to image different parts of the brain. So there's 4 scanners that we have. There's 2 that are like this, horizontal. One is used for humans, and one is used for monkeys by Doris Tsao. And you put a subject on a table here, you put a radio frequency coil around the head, and then they slide into the scanner. And most of the postdoctoral fellows and graduate students that are doing experiments on this are super users. They know how to program up the scanner, and they know how to operate the scanner on their own. And so they can run experiments, not on their own. They have to always have a scanning buddy with them for safety. But they do experiments in the evening, on weekends, and so forth. There's a vertical Bohr magnet that occupies 2 stories that's used by Richard Anderson in the biology division, also for monkey imaging. In his case, he has monkeys that need to move joysticks around, so they need to sit upright and do things. And so they're lowered into this thing with an elevator, and then they sit inside it upright. And then there's one that is a very high field magnet at 7 Tesla. These others are at 3 and at 4.7 Tesla. This one is very strong with a very small bore that's used for rodents, rats and mice. So it has a very small bore. You couldn't fit a human or a monkey into there. And it has an insert here that is used for PET, for positron emission tomography. So you can concurrently obtain very high resolution through MRI imaging and do PET imaging that tells you something about function. This is used a lot for cancer research, actually. So you can have radioactive probes that bind to tumor cells, and those are labeled, and you can visualize those with the PET imaging that's inside here. And then at the same time, you get a very high resolution MR scan of the whole animal, and you superimpose those 2. This is work that's been done by Russ Jacobs and was done by Scott Fraser before he moved to USC. The big questions that the imaging center, so we got a grant from the Moore Foundation 10 years ago that started the imaging center, $28 million to start it up. And that grant and the sort of overarching questions of the imaging center are these are very big questions. How does the brain generate the mind? How do we make decisions? There's lots of people over here who are economists that are studying this, trying to use some of the mathematical tools from economics to ask questions about how those processes are implemented in the brain. And then we study disorders like autism, so we do a lot of our imaging from my lab there as well. So these are just some of the subtopics. One feature that's pretty unique to the Caltech Brain Imaging Center is that it allows comparisons across species. So we are collaborating, for instance, with Doris Tsao, who will be down there and can show you her scanner. She does a lot of fMRI in monkeys, asking how different parts of the monkey brain process faces. She looks at face processing. Well, so does my lab, and we ask the same analogous question in humans. And so you might ask, well, are there differences or similarities between how the monkey brain and the human brain process faces? And you might imagine that, at the level of the retina, at the level of early visual processing, they're identical. Monkeys can see faces perfectly fine. They can tell them apart. But at higher levels, trying to make inferences about what someone is feeling, complex social judgments based on faces, our brains make those, but the monkeys presumably don't. So at some point you would think processing would be the same in monkeys and humans, and at some points it would diverge and there would be uniquely human processes. And we can ask the questions, where in the brain does that happen? And so you slide a person into the scanner and you get images of the brain that look like this. So you get slice by slice. You go through the brain inside the skull here. There's the eyeball appearing and the nose over here. And so out of all of those slices, you can then reconstruct a whole brain, which is some of the pictures that you'll see there. And I think I'm going to skip this next one. Let me just go straight to this. So you are all, in some way, I think, allocated to these groups that are going to be led by these staff people up here. And here are your relative time slots. So these groups 159 over here, you will be going to this particular scanner and working with, or talking with this person here. This is the human scanner. So this stands for Total Imaging Matrix 32, which is just the type of scanner that has a 32-channel head coil for parallel imaging. So this is our human scanner. You will see this here, and Vikram Chib is a postdoc who works on decision making, who can tell you about some of the work that he does. This one here is a monkey scanner that Doris Tsao, who's a professor in biology, uses, her lab uses. Her lab is just adjacent to the scanner. It's very similar, hence the name, to the human scanner. It looks almost the same, but she uses that for monkeys and can tell you about some of her work on face processing in monkeys. This one here, this is our highest field magnet. These groups will be with Mike Tishka, who's associate director of the imaging center, and he can tell you about this scanner. This is the one that has both positron emission tomography and MRI together to image, for instance, tumor movement in rodents. And then over here, the last one is the vertical bore magnet, which is the one that Richard Anderson's lab uses to look at reaching and decision making in monkeys. So that's what's in store for you next. Yeah.
All right, I have a few words.
Take it from there.
Thank you.
And we'll